Okay, last but not least, let's look at specific ways that our bodies and other organisms, how they can defend against infections that are going to affect our homeostasis. So immune system is a body system responsible for defending against infection. And then pathogens are cells that are able to cause disease. They're found in the environment and affect all types of organisms. Plants, invertebrates, and vertebrates have multiple non-specific immune responses. These responses work to defend the organism against any foreign pathogen. These pathogens can include viruses, fungus, bacteria, prion, and toxins. Invertebrate immune systems have non-specific responses and defense mechanisms. Their first barrier to defense is often an exoskeleton or bacteria found in their digestive pathways. Most invertebrates contain hemocytes in their hemolymph, which is blood equivalent. They're able to ingest pathogens via phagocytosis, while others can form capsules around and isolate large parasites. Some invertebrates also display pathogen-specific defense responses. So, for example, sponge cells are able to distinguish pathogenic cells from non-pathogenic, and earthworm cells are able to respond more quickly the second time a pathogen is encountered. Like invertebrates, plants also have entry barriers to prevent infection. Some defenses include thickened cell walls, waxy cuticles, and then stomata control, and bark, of course. Plant defenses against pathogens also include molecular recognition systems with systematic responses. Infections trigger chemical responses that destroy infected and adjacent cells. So here's one example. The recognition of microbial components at the cell's surface triggers the innate immune response. Next, the microbe injects effectors into cell to interfere with the immune response. Plants have a second line of defense and can recognize effector molecules or their effects on the cell. One system uses pattern recognition receptors, or PRR, which detect evolutionary conserved molecular patterns of pathogens. These receptors are embedded in and span the cell membrane. Vertebrate immune systems have non-specific defense mechanisms against pathogens. Our non-specific barriers include skin, mucus, tears, sweat, and hair. So phagocytes are types of white blood cells that are able to move about the capillaries to sites of infection. When pathogens are detected, they non-specifically engulf and digest them with enzymes. And this is really crazy that this can happen. It's amazing. Phagocytes non-specifically engulf pathogens via endocytosis, which is referred to as phagocytosis. So here we basically have some microbe or particle that is just completely being engulfed by the phagocyte. And here at step 4, this is pretty interesting. We have a lysosome that's going to come in and bring an enzyme, or a lot of enzymes, over to that microbe or particle. It's going to help digest that particle, and then all of this excess material is just going to be discharged out. Phagocytes can also initiate a nonspecific inflammatory response when pathogens are detected. This localizes the infection is an attempt to eliminate the pathogen and remove damaged tissue. So here, like let's say you have some sort of an infection, there's all of this bacteria that enters into the open wound. What's going to happen is we're going to get a chemical alarm signal, and through that blood vessel we're going to actually bring over all of these phagocytes, which are going to help eliminate all of the bacteria here. Mammals have specific immune responses that are triggered by pathogens that disrupt homeostasis. A response is initiated after antigens are detected. So we have our innate immunity, which is going to be a rapid response, and adaptive immunity, which is our slow response. And these are all the types of cells that are going to be in each type of response. Pathogens that enter the body initiate an immune response. Proteins and molecules on their surface, called antigens, are recognized as foreign by the body's cells, so we can actually, we're actually able to read all these antigens on the outside and know, okay, that's not part of our body, something has to be done about this. After these antigens have been detected by the white blood cells, two types of responses can be triggered. We can have cell-mediated or we can have humoral. So we'll look at those two types, but know that it's going to be a little different than our innate and adaptive. So once again, how we actually can tell if something is going on in our body is through these antigens. So here's this an image of the antigen presentation. So phagocytosis of the enemy cell, which is our antigen, fusion of the lysosome and phagosome, enzymes start to degrade the enemy cell, broken into small fragments, and released through the surface. So this is an example of our cell-mediated response. Intracellular infections occur when the pathogens infect individual cells, which then display antigens on their surface. Cytotoxic T cells are a type of lymphocytic white blood cell. They target infected cells by binding to them and secreting a protein which destroys them. So, if this is our targeted cell with the antigen in it, our cytotoxic T cell can come in, it can secrete the protein, and then that protein will kill that targeted cell. Once again, that's the cytotoxic T cells. Each T cell targets a specific type of antigen. When they're activated, they release cytokines, which help the activity of other immune cells. These signals affect the humoral response. So our humoral response is when B cells, that are a type of lymphocyte that produce antibodies that bind to specific antigens. These are Y-shaped proteins that are used by the immune system to neutralize pathogens. So once again, this is the antigen here, and then our antibodies, and there's a binding site for them. Each antibody has a region that is specific to an antigen. When an antibody binds to the antigen on a pathogen, it aids in controlling the infection. So here are antigen binding sites. Here's our whole antibody and our antigens binding here. Once again, that is in order to help control the infection. So for a humoral response, B cells for a specific antibody are stimulated to develop by cytokines released by T cells. The antibodies they produce target the pathogenic cells for destruction by other leukocytes. When an antigen is detected, only lymphocytes with reactive receptors are allowed to mature and produce. This is called clonal selection and quickly increases the amount of specific antibodies. After the infection has been dealt with and the antigens are no longer present, the lymphocytes will gradually begin to die. However, some live on as memory cells. These remain inactive until the same pathogen is encountered again. When they quickly reproduce, as a result, they confer an immunity to the pathogen. So that's the reason why for the second time we have exposure to some sort of an infection, why it's so much quicker to respond is because of these memory cells that remember how to make the particular antibody.